Okay, good morning, my dear students. Myself, Kirti Rai, working as JL in Sarville Junior College. Today, we are going to start the chapter Reproductive Health. And in this chapter, we are going to discuss about the very short answer questions, and long answer questions. However, for this chapter, there is no long answer question. And as per the also, only VSAQs, that is very short answer questions, and short answer questions will be coming in the public exam, as well as we'll also discuss about some of the NEET questions. So let's start with the topic reproductive health. So what is reproductive health? Reproductive health refers to having healthy reproductive organism with a normal functioning. So that is the reproductive health, and it is very important for a happy life. So first question is, what in your view are the reasons for population explosion, especially in India? What in your view are the reasons for population explosion in especially in India? The answer for this question is increased healthcare facilities along with better living conditions had an explosive impact on the population growth. So because of increased healthcare facilities, so anyone is falling sick because of the development in the medicine or development in the field of drug production or cure for various diseases, the healthcare facilities have been improved because of the population growth is increasing. Next is, the probable reason for this growth rate are decline in the death rate, as well as maternal mortality rate and infant mortality rate. Means death rate has declined, and more people, maternal mortality rate has also increased, and infant mortality rate has also been increased. Because of that, there is population explosion. So that is the answer for this question. Next question is it true that MTP, what is MTP? MTP is nothing but medical termination of pregnancy. So what is MTP? Medical termination of pregnancy. Is it true that MTP is not meant for population control? Then why did Government of India legalize MTP? So what should be the answer of this question? It is considered to be the only available choice to get rid of unwanted pregnancies. So MTP, medical termination of pregnancy, it is allowed till 24 or 27 weeks of pregnancy. After that, it is not allowed. By legally, it is allowed in India. So it is considered to be the only available choice to get rid of unwanted pregnancies resulted due to casual or unprotected intercourse. So one of the options is that. Second one is failure of contraceptive used during coitus, or in case of rapes, or in case of confirmed incurable genetic disorders in the fetus. So all these are the reasons for which people will be going for termination of the pregnancy. So this is the only available option for all these cases, and mostly it is for the incurable genetic disorders. So in the fetal stage only, by various techniques, either by ultrasound or by amniocentesis or by various other techniques, we can identify the genes of the individual. We can study the chromosomal pattern, we can study the chromosomal structure, and if there are any genetic disorders, genetic disorders which is incurable, which doesn't have any cure or which doesn't have any medicine, so all those things can be confirmed at the earlier embryonic stages only. And if it is of pregnancy could be harmful or even fatal, either to the mother or to the fetus, or for both MTP is inevitable solution. So these two are the things. The third option is if the continuation of pregnancy, because in some cases, due to continuation of the pregnancy, it causes health hazards or it is very harmful to the mother, that is the one who is carrying the baby, or the fetus, or for both. So at that time, MTP, that is medical termination of pregnancy, becomes the inevitable situation solution for the people. So that's why government of India has legalized the MTP. Next question is, mention the advantages of lactational amnesia method. What is lactational amnesia? Lactational amnesia is nothing but when the mother is feeding the baby, during that time there will not be any ovulation. So mention what are the advantages of this lactation. So ovulation generally will not occur during the period of intense lactation by the mother following parturition. That is after the delivery. If the mother is feeding the baby intensely, means continuously feeding of the baby without any other food products, then mostly those people, those mothers, they will not undergo ovulation, means there will not be any menstrual cycle. So because of the lack of ovulation, there will not be any ova generated, and hence it is a preventive measure for pregnancy. So this is known as this period where during the intense lactation, when the mother will not be ovulating, is known as lactational amnesia. Next question is, mention advantages of lactational amnesia method. Okay, that we have discussed. And some couples utilize the contraceptive benefits of these methods. Next question. This is a short answer question. Briefly describe the common sexually transmitted diseases in human beings. So we have to describe here the sexually transmitted diseases in the human being. These are some of the sexually transmitted diseases which are most commonly seen in the human beings. The first one is gonorrhea. This disease, here in this column we have the name of the disease, in the other column we have the causative organism which causes the disease. So gonorrhea is the name of the disease, and it is caused by the organism Neisseria gonorrhea. It's a bacterial disease. The second one is syphilis, which is caused by Treponema pallidum. This is also another sexually transmitted disease. And the next disease is genital herpes, herpes simplex virus. It is caused due to a virus known as herpes simplex virus. The next disease is genital warts or cervical cancer. This is caused by human papilloma virus. This disease is caused by human papilloma virus. The next one is trichomoniasis. Trichomoniasis is a disease, name of the disease, and it is caused by the organism Trichomonas vaginalis. So name of the organism is Trichomonas vaginalis. The another one is chlamydiasis. So the disease is chlamydiasis, and it is caused by the organism Chlamydia trachomatis. And hepatitis B, it is also a viral disease which is caused by HBV, that is hepatitis B virus. And last and not the least one is HIV, that is HIV infection or AIDS, and it is caused by human immunodeficiency virus. So these are some of the most common sexually transmitted diseases in human beings, and mostly it is spread through sexual contact between the infective individuals. Now, what exactly we mean by sexually transmitted diseases which get transmitted through sexual contact? The early symptoms of STDs are minor, that is, sexually transmitted diseases are very minor and include itching or fluid discharge or slight pain or swelling in the genital region. So, these are some of the early symptoms which will be very much minor, but later on it can cause hazardous effects to the individual untreated. If sexually transmitted diseases, um, it should be identified by these above symptoms. If it is not identified and not treated properly, untreated sexually transmitted diseases are major threat to a healthy society. So it may cause various other problems to the individual, and it may also lead, sometimes it may also lead to the infertility or some of the um, highly infectious diseases. Now, how can we prevent the sexually transmitted diseases? It can be prevented by avoiding sex with the unknown partners or multiple partners, and using condoms during the coitus, or consulting qualified doctors for early detection of STD and getting complete treatment in case of infections. So once you identify the minor symptoms or major symptoms, is immediately they should consult the qualified doctor for the early detection of STDs, and they should get complete treatment in case of infection. So it should not be avoided or it should not be neglected. We have to identify the disease and immediately it should be consulted with the doctor, and proper treatment of this infectious disease should be continued till the symptoms are gone. So that's how we can prevent the STD and we can prevent the healthy life. The next question is, describe surgical methods of contraception. So what are the surgical methods of contraception? Surgical procedure in male vasectomy and in female is tubectomy. So these are the two surgical procedures which are seen in the which are the surgical methods of contraception. One is vasectomy, which is done in the male, and second one is tubectomy, which is done in the female. Let's see what exactly they are. Vasectomy. So vasectomy is nothing but a small part of the vas difference on either side of it is removed or tied up through a small incision on the scrotum. This way, sperms are prevented from reaching the seminal vesicle. You can see in this diagram, you can see here, this is the testis and this is the epididymis, and this part, this region is known as, this region which is coming out from the epididymis and going is known as vas difference. So here vas difference is cut or tied in the case of vasectomy. Vas difference is cut or tied. In some cases it is completely cut, and in some cases it is tied, which can be reversed also. So that's vasectomy, which is done in the male individual. So small part of the vas difference on either side is removed or tied up through a small incision on the scrotum, because the vas difference starts from the scrotum, in which the testis is located. This way, sperms are prevented from reaching the seminal vesicle. The next question is tubectomy. Sorry, the next surgical method is tubectomy, which is seen in the female, or which is done in the female. Small part of the fallopian tube or both sides is removed or tied up through a small incision made in the abdomen or through the vagina. So this will block the entry of OI into the fallopian tube. This will block the entry of OI into the fallopian tube, and thus pregnancy is prevented. So here let's see in this diagram, in this picture, let's see. So this part is O, this part is ovary. This is the fallopian tube. So here, what they are doing, they are cutting a small incision. They are making a small incision in the fallopian tube, or they are tying it. And how the small that incision is made? It is made through the um through the vagina or through the abdomen. And once it is tied or once the incision is made or cut the fallopian tube, then the ova which is get coming out from the ovary will not be passed from the fallopian tube. It does not goes, even though it reaches to the fallopian tube, it cannot go to the uterus. So there is no passage of sperm or there is no entry of sperm and ova. So because of that, the fertilization is prevented, and this can be used as a contraceptive method. So that is the answer of this question. Vasectomy and tubectomy. This is the tubectomy picture and this is the vasectomy. The next question. Write short notes on any two of the following. So here you have to write short notes on any two of the following. This is also a short answer question. Let's see the answer. The first one is in vitro fertilization. So the in vitro fertilization we are going to see here first. What is in vitro fertilization and embryo transfer? Fertilization of ovum by sperm, which is done outside the body of a woman, is called in vitro fertilization. So, fertilization of ovum by sperm. Generally, the fertilization of ovum by sperm is done within the or it occurs within the fallopian tube. But here, in the case of in vitro fertilization, it is done outside the body. That's why it is in vitro, and it is called in vitro fertilization. The resultant early embryonic stage. So after the fertilization, the zygote will undergo division, and it will get converted into two-celled, three-celled, or four-celled, and then finally morula and blastula. So, and that blastula stage, or it is also known as early embryonic stage, is transferred into the mother's uterus for further development. So, this embryo is transferred in the intrauterine, that is inside the uterus. So, that is known as in vitro fertilization, embryo transfer. In this method, which is used in the test tube baby procedure, this in vitro fertilization method, we use it in the test tube baby procedure. So here, ova from the mother, ova is collected from the mother or wife, or it can be collected from the female donor. If the mother is not able to produce ova, then it will be collected from the female donor, and sperm can be collected from the husband or male donor if the husband is not able to produce sperm or the sperm count is very low. And then that ova and sperm is mixed and induced to form the zygote under stimulated condition. So in that it is done, this fertilization as it is done outside the organism body. That's why we call it as in vitro fertilization. The uh, in outside the organism body, stimulated conditions are provided, the conditions which are similar to that of the fallopian tube or inside the body, which is suitable for the fertilization. That kind of condition is provided to the uh, for the fertilization of sperm and ova. And after the fertilization, the zygote stage or after the early embryonic stage is transferred to the uterus. So this process is known as in vitro fertilization and embryo transfer. In if the mother's uterus is not medically fit to receive the embryo. Now suppose if the mother uterus is not very well developed, or it is not able to hold the baby, or implantation process is not done properly, then if the mother's uterus is not medically fit, then to receive the embryo produced in in vitro fertilization, it can be implanted to the surrogate mothers also. So surrogate mothers are the biological mother will be the one who is providing the ova, but surrogate mother is the mother who is carrying the baby, because the biological mother is unable to carry the baby because the uterus is not well developed or medically not fit. Then that early embryonic stage will be transferred to the surrogate mother. So that is in vitro fertilization. Next topic is intrauterine devices or IUDs. What is IUDs? Nothing but intrauterine devices. These devices are inserted into the uterus by doctors or trained nurses through the vagina. And different types of IUDs, such as non-medicated IUDs or lip lobes, lip lobes, copper releasing IUDs, which will be releasing example of copper releasing IUDs are copper T or copper 7 or multi load 375, and hormone releasing IUDs, which will be releasing progesterone hormone or LNG20, are available for the contraception purpose. So these are the different types of IUDs. And IUDs promote phagocytosis of sperm by white blood cells within the uterus. So these IUDs, intrauterine devices, are inserted inside the uterus. It is inserted inside the uterus, and what they do is they will avoid the and they will avoid the process of fertilization by phagocytosis of the sperm. Either they will be killing the sperm or by inactivating the sperm, these intrauterine devices, they will inactivate or they will kill the sperm, and ultimately the sperm will be phagocytized by the white blood cells which are already there in the body. So that's how when the sperms are not active or they are not alive, then obviously they cannot fertilize the ova. So by this technique, this is one of the contraceptive method which is used worldwide. The next is, you can see here some of the images of copper IUD or strings which is inserted inside the uterus. Next is intracytoplasmic sperm injection. The next one, C option, was intracytoplasmic sperm injection or ICSI. So what is this procedure? In this procedure, sperm is directly injected into the ovum with the help of a microscopic needle to form an embryo in the laboratory. So here what we have seen earlier in the test tube baby or in vitro fertilization, just the ova and the sperm are kept in the suitable condition and they themselves will fertilize. But in this intracytoplasmic sperm injection, the ova, inside the ova, directly sperm is injected inside the ova through the needle. Okay, so through the microscopic needle, the embryo is formed by inserting the nucleus of the uh, sperm, and it is done in the laboratory. Now later, once the zygote is formed, that is after the fertilization of sperm and ova, this embryo will be later on transferred to the uterus or fallopian tube for the further development. So that process is known as intracytoplasmic. Intra means within, inside. Cytoplasm, inside the cytoplasm, the sperm injection is taken. So inside the cytoplasm of the ova, sperm is injected. That's why it is known as ICSI. This method is employed to assist. This method is employed to assist the couples where there are problems with the sperm, such as decrease in sperm count. So because of this decrease in the sperm count, or the sperms are not highly motile, which are not able to reach to the ova or not able to fertilize the ova, this technique is used. So especially this technique is used in case of male infertility. Directly the ova is collected from the mother and sperm is collected from the father, and it is injected inside the cytoplasm through the injection. So that is intracytoplasmic sperm injection. The next question is, suggest some methods to assist infertile couples to have children. So in some cases where the couples, where the people cannot, due to many problems, if those people are not able to conceive the child, then what are the different methods which can be used to assist them to have children? So one is we have already seen in vitro fertilization and embryo transfer. So in vitro fertilization and embryo transfer. Here ovum by sperm done, ovum and sperm, fusion of ovum and sperm is done outside the body of a woman and is called in vitro fertilization. The resultant early embryonic stage is transferred into the mother's uterus for the further development. Next one is assisted reproductive technology. In the cases where such corrections are not possible, the couple could be assisted to have children through certain special techniques. These techniques are known as assisted reproductive technology. The following are some of the important techniques: in vitro fertilization and embryo transfer, zygote intrafallopian transfer, gamete intrafallopian transfer, or intracytoplasmic sperm injection, which we have already seen. So already we have seen the in vitro fertilization. Let's see zygote intrafallopian transfer. In the zygote intrafallopian transfer, this is another technique to overcome the infertility. The ovum is extracted and is fertilized in vitro, and the zygote is transferred to the woman's fallopian tube. So in the in vitro fertilization or embryo transfer, we have seen that the ova and sperm is fertilized. They are outside the body, right? In the test tube, we can say in the stimulated condition. But here we are extracting. Here the ovum is extracted and fertilized in vitro, and zygote is transferred to the woman's fallopian tube to complete its further course of development. There, at the embryonic stage, that is at the blastomere stage, we are transferring the embryo into the uterus. Here, the embryo is transferred into the fallopian tube, and here the zygote stage only, that is one cell, single cell stage, is only transferred into the fallopian tube. And after that, the next course of development will be taking place inside the fallopian tube, and then from there it goes to the uterus. So just at the zygote stage only, we are transferring the uh, zygote directly into the fallopian tube. The next one is gamete intrafallopian transfer. So here gamete itself is transferred. Some women cannot produce ova, okay, but they are okay. Some women cannot produce ova either due to defects of or diseases in the ovaries, but they can still provide suitable environment for fertilization and further development of the embryo. So the only problem they are facing is they are not able to produce the ova. So what they do is they will be taking, in such cases, an ovum collected from a donor. An ovum is collected from the donor and is transferred to the fallopian tube of the recipient woman for the process of fertilization. So the ovum is collected from the donor because biological mother is not able to the biological mother is not able to produce the ova. It is collected from the donor, and that ova is only transferred into the fallopian tube. So here there is no fertilization. The ova is at the ova stage only. They are transferring it into the fallopian tube, not the zygote, not the embryo. So that's how this gamete intrafallopian transfer is done, and how that's how it is different from the zygote intrafallopian transfer or the in vitro fertilization or embryo transfer. And then this method is known as GIFT. The last one is artificial insemination. In the artificial insemination, where in case this is seen in infertility either due to inability of male partner to inseminate the female or due to low sperm count. So this is mostly done when there is a infertility problem with the male. So if it is due to insemination, means he is not able to inseminate, or due to the low sperm count. So artificial insemination technique is suggested in these cases. In this technique, the semen is collected from the husband or healthy donor, and it is introduced into the uterus for achieving the fertilization. So here either the semen can be collected from the husband itself, or or it can be collected from the healthy donor, and then it is introduced into the uterus. So here we are not introducing ova or zygote or the embryo. Here directly we are introducing only these sperms into the uterus, and then that sperm will swim and it will be reaching to the fallopian tube and it fertilizes the ova. That technique is known as artificial insemination because it is not a natural insemination. Artificially it is done. That's why its technique is known as AI or artificial insemination. So those were some of the reproductive assisted technology for the infertile couples. The next is, what is amniocentesis? What is amniocentesis? Name some of the disorders that can be detected by amniocentesis. So amniocentesis is a diagnostic procedure. It is a diagnostic procedure to detect genetic defects in the unborn baby. So in the unborn baby, that is in the fetal stage or embryonic stage only, we can identify the genetic defects, and by this technique of amniocentesis. So in this procedure, usually a needle is inserted through the mother's abdominal wall into amniotic sac, and amniotic fluid is collected. So here the needle is inserted through the mother's abdominal wall and amniotic sac. Amniotic sac is nothing but there are four extra embryonic layers around the embryo, and one of the extra embryonic layer is known as amnion. And around this amnion is filled by amniotic fluid, which is covered by amniotic sac. This amniotic fluid will be collected in the technique of amniocentesis. The chromosome of the stained fetal cells. So after collecting the amniotic fluid, they also consist of those amniotic fluid also consist of some of the those amniotic fluid also consist of the chromosomes which are present in the fetal body. So those chromosomes will be stained and fetal cells are examined under the microscope for any abnormalities. So those abnormalities can be identified by the technique of amniocentesis. The most common abnormality that can be detected is Down syndrome. So some of the genetic disorders can be identified, and some of the Down syndromes, that is Down syndrome, Edwards syndrome, and Turner syndrome. These are the most common syndromes which can be easily detected by the amniocentesis technique. And by this amniocentesis technique, it can be early detection of the embryo can be done, and if it can be the fetus can be removed, or it can be used for medical termination of pregnancy can be done by identifying the genetic diseases. So that's why this technique is very much popular, and it is most commonly used for the identification of the genetic disorders. But in some cases, this technique is also being misused, and it leads to the female feticides. And that's why at some places, most of the places, this technique has been banned. Now let's see the questions on NEET from this chapter. Some of the questions which are asked in the NEET. The first question is, the functions of copper ions in copper releasing Iuds is. So what is the functions of copper ions which is released from the IUD, that is copper T, which is an intrauterine device. So what do? They inhibit, first option is they inhibit gametogenesis. That is, they will not allow the process of ovulation or the process of spermatogenesis. Then the second one is they inhibit ovulation. Then they suppress sperm motility and fertilizing capacity of sperms, or all of the above. So what could be the answer of this question? So in the intrauterine devices, we have already learned that what they do is they will be making the sperm immotile, or they will be making the sperm either they kill the sperm, or they will decrease the motility of the sperm, and then later on they will be phagocytosed by the WBC. So the answer of the question is C, that is they suppress sperm motility and fertilizing capacity of the sperm by making them inactive. So other options are not correct for this answer. So the answer is the copper ion in the copper releasing Iuds, they will be suppressing the sperm motility and fertilizing capacity of the sperm. The next question is, in case of a couple where the male is having a very low sperm count, which technique will be suitable for the fertilization? And this question was asked in the year 2017 in the NEET exam. So in case of couple where male is having very low sperm count, just now we have seen artificial insemination technique, right? In which the male was in which if there is a low sperm count, then the uh, sperm will be collected from the husband or it will be collected from the male donor, and then it will be introduced inside the uterus. So what will be the answer of this question? The answer of this question will be artificial insemination. Intrauterine transfer is done. Intrauterine transfer is done in the embryo, where the embryonic stages in the in vitro fertilization, embryonic stage at the blastocyst stage is transferred into the uterus. So that process is known as intrauterine transfer, which is mostly seen if there is a problem with the fertilization. And then gamete intracytoplasmic fallopian transfer, mostly it is also done in here. The gamete is transferred into the fallopian tube. So this technique is also not seen in the then none of the above. So the answer of this question is artificial insemination, where the male is having very low sperm count, and this technique is suitable for that. The next question, which of the following is incorrect regarding vasectomy? So what is vasectomy? We have seen. Let's see the options. The vas difference is cut and tied, no sperm count in the seminal fluid, irreversible sterility, or no sperm occurs in the epididymis. So what is incorrect regarding the vasectomy? So the vas difference is cut and tied. Yes, it is cut and tied. So that is the correct one. No sperm count in the seminal fluid. Obviously, because the seminal fluid will not be having any sperm, because the vas difference is cut and tied. Irreversible sterility. Yes, sometimes because some in the cases where it is tied, so we can reverse it back by removing that tied part. So again, it will, if it is cut, then we can't, but if it is tied, we can reverse it. So this is also correct. No sperm occurs in the epididymis. No, this will not be the correct one, because the testis will be producing the sperm. The only difference here occurs is the sperm is not passed to the vas difference, and it will not reach to the seminal fluid or urethra, or the sperm will not reach to the uh, urethra and then to the penis, but there will be production of seminal fluid, there will be production of prostate glands, everything will be produced. The only thing is there will not be sperm. But sperm will be reaching to the epididymis. From epididymis, the next part of the epididymis is vas difference. This vas difference is cut and tied. So this is the correct answer of this question. That is, the incorrect statement is no sperm occurs in the epididymis. This is the incorrect statement. The testis will be producing the sperm, and sperms will be there in the epididymis, but sperm does not reaches to the urethra because there is a cut or incision made in the vas difference. So the correct answer is no sperm occurs in the epididymis, which is an incorrect statement regarding the vasectomy. The next question, which of the following is a hormone releasing IUD? IUD is nothing but intrauterine devices, and this question was asked in 2016. So loops, no, it does not releases the hormones. Multiload 375, this also does not release hormone. Copper 7, it releases copper ion, so that also does not. So LNG is the correct answer. LNG 20 is a type of intrauterine devices which will be releasing the hormone, and the hormones released from LNG 20 or intrauterine devices will be mostly the progesterone, combination of progesterone and estrogen hormones. So the answer of that question is the IUD, which is releasing hormone, is LNG20. The next one is embryo with more than 16 blastomeres formed due to in vitro fertilization is transferred into what? So once the in the in vitro fertilization, once the fertilization is done, then the embryo at the 16 blastomere stage, that is blastocyst stage, it is transferred into the in vitro it is transferred into the body of the female. So where it is transferred, in which part it is transferred, we have to identify in this answer. We have to find out the answer. So at the 16 blastomere stage, is it transferred into the fallopian tube? No, it is not transferred into the fallopian tube. In the fallopian tube, gamete stage is transferred, that is ova or sperm is transferred, that becomes gamete intrafallopian transfer. Fimbriae is a part of the fallopian tube, so that also cannot be the answer. Cervix, it cannot be transferred into the cervix because cervix is not a place where the implantation of the embryo occurs. So the 16 blastomere stage is transferred into the uterus because uterus is the place where the implantation of the embryo at the blastocyst stage will be occurring. So the 16 blastomeres formed due to in vitro fertilization is transferred into the uterus because this is the place where implantation of the embryo will be taking place. The next question is, in context of amniocentesis, which of the following statement is incorrect? So what is amniocentesis? We have already seen. Let's see which of the options is incorrect related to the amniocentesis technique. So the first one is usually done when the woman is between 14 or 16 weeks pregnant. Option B is can be used to detect Down syndrome, can be used for prenatal sex determination, can be used for detection of cleft palate. So which one is incorrect statement? So let's see the first option. Usually done when the woman is 14 to 16 weeks pregnant. That is the fourth month usually done. Yes, it is the correct one. The next one is can be used to detect Down syndrome. Yes. First of all, what is amniocentesis? Where an amniotic fluid is collected from the amniotic sac, right? That amniotic fluid contains the chromosomes, right? Those chromosomes are the part of the fetal body only. So we can identify that chromosomes, we can study the karyotype of those chromosomes, and we can see if there are any genetic disorders or what is the number of chromosome. We can identify any type of syndrome. So the second option is can be used to detect Down syndrome. Yes, it is correct. Can be used for prenatal sex determination. So we can also determine the sex of an individual by identifying the chromosomes. If it is XX, then the embryo is female. If it is XY, then the embryo is male. So yes, it can be also used by, it is also used for prenatal sex determination. Can be used for detection of cleft palate. So what is cleft palate? Cleft palate is nothing but the lips. Generally, if there is a gap in the lip, that is upper lip is not completely formed. There is gap in the upper lip because of incomplete formation of the limb due to some of the inherited problem. So this cannot be done. This cannot be detected by the amniocentesis because in the amniocentesis technique, mostly here we identify the chromosomal pattern. So by this we can identify any of the genetic defects, but we cannot identify the cleft palate, which will be mostly seen in the 5th month, and it can be detected only in the ultrasound. So the answer, the incorrect statement is it can be used for the detection of cleft palate. So that is the answer of the question. So amniocentesis technique cannot be used for the detection of cleft palate in the growing fetus. Next one is hysterectomy is the surgical removal of, which was asked in the year 2015. Mammary glands, vas difference, uterus or prostate gland. So hysterectomy, mammary glands, no, it is not the correct one. Vas difference, that technique is vasectomy. Prostate gland also cannot be. It is the technique which is used for the surgical removal of the uterus. Here, if there is any infection or if due to any cyst development in the uterus, this technique is mostly followed where the completely uterus is removed from the female body. So the technique is known as hysterectomy. The other techniques are vasectomy, which is in which the vas difference is cut or tied, or the tubectomy in which oviduct in the female, it is done in the female, oviduct is cut or tied. The next question is, which of the following is not a sexually transmitted disease? So out of these, which one is not a sexually transmitted disease? Encephalitis, syphilis, AIDS, or gonorrhea. In the sexually transmitted diseases, we have seen all these. We have seen syphilis, so it is a sexually transmitted disease. We have also seen AIDS, so it is also a sexually transmitted disease. We have also seen gonorrhea, so that is also sexually transmitted disease. Only out of these, the only disease which is not sexually transmitted is encephalitis. So the correct answer is encephalitis. Next question is, tubectomy is a method of sterilization in which? So tubectomy is a technique of sterilization in which of the in which this question was asked in the year 2014, and the uterus is removed surgically. So tubectomy is method of sterilization in. So in the tubectomy, uterus is removed surgically. No, it is the wrong answer. Small part of fallopian tube is removed or tied up. Tubes are inserted into the uterus. Ovaries are removed. So here the correct answer is small part of the fallopian tube is removed or tied, or cut is made in the fallopian tube so that it will not allow the ova to pass to the uterus. And the next one is, tubes are inserted into the uterus. No, that is not the correct answer. Or ovaries are removed. So the correct answer is small part of the fallopian tube is removed or tied up. The next question is, which of the following cannot be detected in a developing fetus by amniocentesis technique? So which of the following cannot be detected in developing fetus by amniocentesis? We can detect various type of syndromes, and we can also detect various type of genetic disorders, but we cannot detect the uh, infectious diseases. So we cannot detect jaundice, we can detect Down syndrome, we can detect Klinefelter syndrome, we can also detect the sex of the fetus. So the correct answer is jaundice, which cannot be detected by the amniocentesis technique because it is the infectious disease. It cannot be identified by it cannot be identified by reading the chromosome structure. So that is the answer of this question. Now let's see some of the common topics of reproductive health. What is the need for reproductive health? Reproductive health problems remain the leading cause of death and disease in the people of reproductive age worldwide. In India, maternal mortality rate and infant mortality rate are very high. Spread of sexually transmitted diseases is still a major problem. Approximately 2 million people in India live with HIV or AIDS. Contraception, the intentional prevention of conception, fertilization of an egg by sperm at the beginning of pregnancy by natural or artificial means, is known as contraception. The contraception prevents pregnancy by interfering with the normal process of ovulation, fertilization, and implantation. So we have already seen different types of contraceptive methods which are used, and there are different uh, techniques which are being used nowadays. So with this, we have completed the VSAQs and SAQs of this chapter, and here this as well as the neat questions of this chapter. So I wish you all the very best for your exams. And for this, the complete chapter has been explained in the webinar class already. So if you want, you can go to that link and you can listen to the full class of this chapter. And in this session, we have only discussed the VSAQs, SAQs, and some of the neat questions which are related to this chapter. So thank you and all the best for your exams. Sir, the class is completed. Hello? The class is completed, sir. You can end the session. Sir, you can end the session. Hello. Sir, end the session.